Hi, everyone. Welcome to Being Patient Brain Talks. I'm Deborah Kahn, founder of Being Patient. One of our missions at Being Patient is really to give people impacted by Alzheimer's better access to the science, a better explanation of treatments and the search for a cure. So today I'm very pleased to have with us Remi Barbier. He is the CEO of Cassava Sciences. Cassava has recently been in the news because they had some promising data in a phase 2 trial for an Alzheimer's drug that is currently under clinical trials. So Remi, thank you so much for joining us.
Thank you for inviting me to present. Remi Barbier, President and CEO, and also founder of Cassava Sciences. And just a quick 10-second introduction. At Cassava Sciences, we have one mission and only one mission, and that is to defeat Alzheimer's disease. We're a biotech company. We have a treatment, an investigational treatment, I might add, for patients with Alzheimer's disease. And we also have an investigational blood-based diagnostic to detect Alzheimer's disease. That's all we do.
Well, you'll have a lot of support on within this community of people impacted by Alzheimer's. We want nothing more than someone to find a better treatment and cure. And so tell us a little bit. First, I just want to start with the very basics. I know you mentioned both. There's a diagnostic and also a drug that you're currently testing. Let's start with the drug, which interestingly enough is both aimed to treat neurodegeneration and neuroinflammation. So tell us a little bit about how that's possible.
Okay, so it's a great question and really a great opening question because you're correct. Normally, a drug aims to do one thing and only one thing. If you have a headache, you take a Tylenol. If you have this, you take that. Our drug, and that's well understood why those things exist, but our drug is a little bit different. We think of Alzheimer's as multiple cascades. It's not a single event. It's multiple things, multiple biological processes happening at once. So, if I may draw an analogy, it's not like, it's not quite a single waterfall. It's more like Niagara Falls, where you've got one big waterfalls, but then you've got all these different waterfalls coming from the left, from the right. Unfortunately, this is what happens in Alzheimer's disease. It's a series of cascades. None of them are good, and the outcome, unfortunately, is well known. So our thesis is that if you go upstream of all of these different cascades, you've got a shot at not just fixing one waterfall, but a number of them. And that's been our basic hypothesis since we started working on our drug almost a dozen years ago.
So let me just so I make sure I'm understanding this correctly. I mean, the presumed pathology of Alzheimer's has been plaques and then tangles and then the inflammation. And there's some scientists who believe if you prevent, even if you have these hallmarks, if you prevent inflammation, then you may not see a symptom of Alzheimer's. Is this the presumption of Simufilam?
I would say, I think what you just said is, it's not wrong, but I would say it's a little simple compared to the full richness and complexity of the disease, because it truly is a mind-boggling, complex disease. And by the way, we're not saying we understand it, in more detail than anyone else. What we are saying is that we have some insights into the biology of the disease that are perhaps a little bit different from what others have been following. But it's that difference that I think results in the type of data that we're seeing, the early evidence for improvement in cognition, the lack of any safety issues, the tolerance, the ability of our patients to tolerate our drug without severe side effects.
So, okay, we're getting a lot of questions in right now, and I want to address one because I think this is the right opportunity to do so. We all know anyone who has a loved one with Alzheimer's, there's only 2 or 3 drugs that are typically from, donepezil, Aricept, Alexon, those type of drugs that really don't. I mean, from my personal experience, I haven't seen them do great things. Maybe, and actually, I was told that they're actually really to be used in the short term. They're not a long-term solution, although people stay on them for a long time just because there's nothing else.
That's right.
So how is and it's the drug that you are studying, it's called Simufilam. Did I pronounce it correctly?
It is a tongue twister of a name.
I actually thought if I'm going to do another business, I'm going to name drugs for drug companies because they're always so hard to pronounce and remember.
No, but Deborah, let me tell you a little secret. We get to invent our drugs, but we do not get to name them.
Oh, interesting.
Yes, we provide the chemical formula to an agency that's part of WHO, and they do whatever they do. They look at it, sideways, upside down, and they say, aha, your drug should be called Simufilam. And you're like, where did that come from? Well, okay. Anyway, Simufilam is the chemical name. Obviously, it will have a more pronounceable, hopefully more memorable marketing name if and when approved. I'm sorry, now we lost.
So the question is, how does it differ from what's currently on which are, I can't even say the word, cholinase inhibitors, right? Did I say that right?
Yes. So there is, I believe at this point, there are about 5, maybe 6 drugs approved for the treatment of Alzheimer's disease. And all of these drugs essentially work in the same manner. You have what are called acetylcholine receptors in the brain. Healthy subjects do, everyone does. And what these drugs do, what these Aricept and that whole, all of its cousins do, is they keep hitting that same receptor over and over and over again, day and night, 24/7, which is not something you want to do to receptors. In any event, that continuous hitting of the acetylcholine esterase of the acetylcholine receptor actually does boost memory. It does boost cognition. That's the good news. The bad news is, to your point, it rarely lasts more than a few months. So let me summarize that thought by saying saying something similar but different. To our knowledge, we have never seen a drug, never in any type of study design, actually improve cognition over beyond 6 to 9 months maximum.
So what about the drug Simufilam? Is that meant to last longer, or is it also?
Yes.
Okay.
Yes. We actually have an ongoing open label study right now. We actually just increased the target enrollment. We were going to recruit 100 patients. We are so overwhelmed with demand that we're increasing it to 150 patients.
And is this still part of the phase 2?
Yeah, it's an open label safety study. So yes, it is not part of phase 3. In any event, what we saw in an interim analysis, in other words, at the halfway point, so 50 patients on our drug for 6 months, what we saw is an increase in cognition and an increase in, or I should say, decrease in Alzheimer's related behavior, which is pretty significant. It was about approximately 10% increase. May not sound like much, but I think as anyone who, who's dealing with patients with Alzheimer's, a 10% increase, a 10% boost means not just the boost, but more importantly, they're not going downhill over that period of time.
So I think one of the things before you experience Alzheimer's, you don't realize how complex a disease is in the sense that it's not just memory loss. In fact, I've come to believe that the memory loss is the easier part to deal with when you're caring for someone. It's complicated in, the behavioral aspects. What did you see with patients? I know, it's early days, still 50 patients, but did it alleviate other symptoms aside from just the memory loss when you talk about behaviors?
Yes. So we used two distinct scales. One called ADAS-Cog, and that's a well-known scale of cognition. The other scale is what's called the Neuropsychiatric Inventory, fancy word, NPI is what we call it. And the NPI measured things that are typically associated, things that probably come to mind when you think of Alzheimer's type behavior. Did I remember to take a shower? Did I remember to change my clothes? Did I remember to eat my meals? Did I have delusions? Did I have psychosis? That's more what the NPI measures. And both cognition and neuropsychiatric function are equally important for these reasons. But to get back to something you said, I agree. It may not be cognition that's the real hit in Alzheimer's. To me, probably the biggest hit is the dignity to the patient. That's the first thing that goes out.
Absolutely. So we're getting a lot of questions, of course, around the trials that you're currently running. And a lot of people are asking whether you'll apply for a breakthrough therapy designation from the FDA. But let's back up a little bit and tell us, I know obviously you have to get FDA approval, right, before you could advance the study. Yes. Where are you at right now? And what's the next step to all of this?
Yeah, so the good news is we just had a big meeting with the FDA in January. We just received the final official minutes of that meeting. So now it's official. And they did give us the green light to move forward with large pivotal phase 3 program. We plan to initiate those 2 phase 3 studies second half of this year. Why the lag? Simply because it takes time. These are very large, complex studies, and you need a lot of drug. You just physically need drug supply, and you need a lot of clinical sites. So it just takes, it's a matter of months to get the organization up and running, to get the drug supply up and running, so that when we start our phase 3 studies, we can just roll right through them.
And how many, it's 1,000 plus, how many will be in phase 3?
So 2 separate studies. One will be approximately 1,000 plus patients with mild to moderate Alzheimer's disease. The second study will be shorter and smaller. It'll be approximately 5, probably more like 600 patients with mild to moderate Alzheimer's disease. And we'll track them for approximately 9 to 12 months.
And, I'm assuming placebo will be in there as well, right?
It's an evil necessity.
Yes. But will the people who enroll in the study be offered the medication depending on the efficacy after they participate?
Yes, we do plan in order to, thank the general public and the patients and the physicians. We do plan to offer open label Simufilam for those who complete the study.
Okay, now I'm going to go back to the basics again, because I think there's a lot of confusion for the non-scientific community. It's a small molecule drug, right? So is it going to be a pill? What's it going to be? And what exactly does that mean when you say we're developing a small molecule drug?
Good question. It's a little bit of shop talk. A small molecule refers literally to the size of a molecule. Because it's a small molecule, it will be a pill. It's a tablet, actually, small tablet. The hardest part will be probably to make sure you remember to take it twice a day, as opposed to perhaps some of the anti-amyloid drugs that are out there where you have to come in once a month or once a week and actually go through a physical exam and an injection and so forth. Ours is, I have to say, it's a little more convenient than some of the other experimental drug candidates out there.
Yeah, and we were getting a lot of questions around this too. And one of the questions was, whether or not you're going to be giving people like spinal taps or PET scans to measure, the amyloid plaque and tangles. Yeah. If you're targeting neuroinflammation, though, I don't know, is that important?
Well, remember, we're targeting neurodegeneration and neuroinflammation, not one, not the other, both. That makes all the difference in the world. So in our early studies, yes, we did require spinal tap. Informally, it's called a spinal tap, where essentially the physician goes in and takes out a very, very small quantity of what's called cerebral spinal, CSF fluid, the fluid that runs up and down the spine. And that's necessary for a couple of reasons. One is you want to make sure that the drug actually reaches the intended target, and it does. We've measured that. But more importantly, we also want to see the drug's effects on all these various biomarkers of disease, things with complicated names like neurofilament light chain, neurogranin, amyloid beta 42, A beta 42, p-tau1. You may have heard all these different terms. Essentially, all these are biomarkers of disease, biomarkers of inflammation. One thing we know for sure with 100% certainty is that if these biomarkers go in this direction, in the up direction, cognition goes in the opposite direction. So we're taking the other side of that bet and saying, look, we have a technique to bring down all of these biomarkers of disease. We think that by suppressing these biomarkers of disease, we're targeting the actual disease itself, the underlying disease, and we can actually improve cognition. And if not improve, certainly stabilize. And that's another point to consider. Stabilizing cognition is a huge win in Alzheimer's disease. Why is it huge? Because we know, once again, with 100% certainty, we know that cognition declines typically by about 4 to 6 points on the ADAS-Cog scale. Cognition declines. It just does in this disease. That's one of the horrors of the disease.
So I'm assuming that, do people have to have the presence of plaque in their brains before they qualify for this study? Do you need to see beta amyloid plaque or tangles in the brain?
Okay, so let me be clear. So in our early study, we had to get CSF fluid. It was a must because we wanted to see if the drug had all the intended effects. All the effects we saw in mice, all the effects we saw in postmortem tissue, all the effects predicted by the science, we had to be 100% sure that the effects were actually in place. We are now sure that the effects are in place. So for our phase 3, we will not require the spinal tap. There'll be a small subset of patients, but very small as a percentage of the total patient population.
So let me just ask you it this way. I mean, it is complex, and I get what you're saying. Like, you have to kind of tackle a multi-dimensional approach. But presumably, if you already have the presence of plaque or tangles in your brain, this drug is not going to reverse that. But what you're saying is possibly it can slow down the deterioration. Is that correct?
Well, again, not to use the same word over and over again, but it really is a complex disease. Let me give you an example why. Every patient with Alzheimer's disease has amyloid plaque in his or her brain, but not everyone with amyloid plaque in the brain has Alzheimer's disease. How's that for complications? So this is why you can't necessarily hang your hat on amyloid plaque and its removal. I think amyloid, the anti-amyloid hypothesis definitely has a place, a significant role to play in the overall science, but there has to be a little bit more to it than just, oh, the patient has amyloid plaque, therefore has Alzheimer's. No, that's not true.
We actually had a question earlier that was sent to us wondering if you're going, would this drug work for other dementias? This person named in particular FTD, but we know there's a lot of different types, vascular, Lewy body, et cetera. Do we know, or are you going to test it on other dementias?
Well, the short answer is we don't know. As scientists, the word no has a lot of importance. So no, we don't know. But is there a good hypothesis? Is there a good reason to believe that our drug can work with multiple forms of neuroinflammation, multiple forms of neurodegeneration? Absolutely, yes. We are a small company, so we're really trying to focus where we think we can have the biggest benefit for the most patients in the shortest amount of time. And to us right now, that's mild to moderate Alzheimer's disease. But yes, with time, we do plan to explore some other indications such as FTD, perhaps Parkinson's, perhaps other forms of neurodegenerations.
And someone is asking, what's a disease-modifying therapy versus symptomatic treatment? What's the difference?
So you take the case of Aricept, okay? So we all agree, I think we can all agree, that you give Aricept to a patient with Alzheimer's and initially the patient gets better. You get symptomatic improvement. The symptoms seem to go away. Unfortunately, with Aricept, as we've said, the symptoms come back with a vengeance. That is symptomatic relief. It's almost like taking a painkiller when you're in pain. You have a migraine or headache, you take the painkiller, the headache doesn't really go away. It's in the background, but you can deal with it. Symptomatic improvement is you make the headache go away. You hit something with the disease that modifies the actual disease itself. So you're no longer trying to put a Band-Aid on it. You're actually trying to treat the underlying condition. That's disease modifying. And that's what we think our drug can do. Except we also know that because of what we've seen in the early studies, we can both improve patients in the short term as well as modify the course of the disease.
Okay, and I guess people really want to know if you're going to apply for breakthrough therapy designation from the FDA for expedited development and a review of the drug. And actually, we got this fair point in earlier that, wow, we developed a vaccine for COVID in record amount of time. What about in this case? Any chance if you're getting early signs that, positive data around this? I mean, obviously you still have a phase 3 trial to run with a lot more people. But, what's the timeline for this? Could it be considered breakthrough therapy?
How should I answer? We're a public company, so I have to be a little bit careful on what I say on, kind of a live show. But the short version is yes, yes, and yes. We're very, very conscious. More than anyone, I think we've been focused on Alzheimer's 24/7 for about 12 years now. We know the damage it can do, and the FDA has told us they're working with us, and they will continue to work with us to make this available to patients as soon as it's proven its paces.
Okay, let's talk a little bit about the phase 3 trial. You told us the approximate timeline for it. What about the early onset folks? Are they included in this, or do you have age restrictions? Who can apply to sign up for the trial?
Yeah, we're still actually putting together the list of what they call inclusion criteria, which is what you call the restriction. It's basically who can and who cannot. But typically, the patients would be between the ages of, say, 50, perhaps 55, and perhaps 80, 85. One, I have to say up front, group of patients that unfortunately we cannot help are those with severe Alzheimer's disease. At that point, the damage is so far out there, so far done that frankly, I think it may be a while before anyone can really help severe Alzheimer's.
So can you define that for, I mean, I know that sounds like a really simple question to ask, but I know within my own family, we constantly go back and forth about what stage my mom's at, because some days are better than others. Yeah. So what is the difference between, like in terms of research, between moderate and severe?
So it's a fair point, and you are absolutely right. And you should go back and forth on a day-to-day basis, because you're right. Some days the patient sleeps very well, feels good, and they're almost, not quite, but almost back to normal. And then unfortunately, some days you're just not there. That is typically mild to moderate. Severe Alzheimer's patients are those who they wake up, they don't know where they are, they don't recognize anyone, they don't know if they've eaten, they don't know if they've showered. It's kind of end stage Alzheimer's disease. At that point, there's no debate anymore. Are we having a good day or a bad day? it is what it is at that point. But as long as you're having that debate with yourself and your family and your caregivers, is today a good day or a bad day? To me, that's a good sign, because you're still in that zone of possible treatment, possibilities of perhaps having more good days than bad days. We can't know, no, there's no silver bullet, and I don't think there will ever be a silver bullet for Alzheimer's disease. But what we're trying to do is put more good days in the good day bucket and minimize the bad day bucket.
So a lot of people describe, have come to the belief, scientists have come to the belief that a treatment for Alzheimer's will look more like one that is used for HIV. So it sounds like that's what you're saying as well. It's such a complex disease. It's not one pill that's going to just cure Alzheimer's. What we have to do is we have to treat some of the biomarkers or the signs that show us that we're on the road to Alzheimer's. So in the case of the drug that Cassava Sciences is developing, I know it's neurodegeneration and neuroinflammation, but is that it? I mean, it's not that that's not enough, of course, but that's a lot. But is it really, when we look at those biomarkers, are we looking for also, I mean, the inflammatory part of this is obviously a very important trigger in this whole cascading process, right?
Yeah. So you're correct. I think it will end up more like HIV and perhaps other fields, where once diagnosed, you will probably see the patient be given a cocktail of drugs, depending on the patient. What biomarkers don't predict with 100% accuracy, or perhaps with any accuracy, is behavior. And as you know, behavior is a big part of Alzheimer's disease. So while we may be able to suppress some of these biomarkers, a lot of these biomarkers, and suppress neuroinflammation and make a real impact on disease progression, certain patients may still need, for example, an antidepressant, or certain patients may need anti-psychosis medication, or anti-this or anti-that, or, so there's room for many, many different players. Again, there will not be a silver bullet for Alzheimer's disease. There'll be room for the anti-amyloid drugs. We're certainly hoping that the Biogen drug gets approved. We hope the FDA does the right thing and provides it to patients. And if so, I think our drug with the Biogen drug, with perhaps a number of other drug candidates out there, hopefully, again, will make a difference in the lives of these patients. And these patients can have more good days than bad days.
And just for the background, the Biogen drug that you're talking about is aducanumab, which is basically targeting the beta amyloid plaque, right? It's an immunotherapy that is going for the plaque in the brain. So you're saying in combination with your drug and a drug like that, it would be a good way to tackle this. It would offer us other options that aren't currently available.
Yeah, the Biogen drug, we call it ADU for short, because the real name is just too long. But the ADU drug has shown some promising effects Unfortunately, those effects were found in kind of a minority of patients. You know, you have to be fair and you have to really, you know, be rational when you look at the data. They did two very, very well-controlled, very well-designed studies, large, complicated, long-term studies. The drug did not work in one, but the drug seemed to have, the high dose in particular, seems to have a real effect in some patients some of the time. You know what? These patients have nothing all the time. Why not provide them with a drug that might actually help them? And that's our rationale for its approval. Likewise, I would not be happy if our drug only worked for a minority of patients, and I don't think it will. I think it will work for a majority of well-diagnosed patients with Alzheimer's disease. This is why I say perhaps, you know, our drug with the ADU, with a couple of others, can provide a number of good days for these patients who don't have that many good days.
So when you say the majority, like what would make this a successful clinical phase 3 trial? Like what percentage of people do you have to see having a positive impact?
I wouldn't say the number of people. I would say what impact does it have? I would start with the impact and then back into the people, the number of people. I tell you, I think if we can show a 10% difference or a 10%, you know, a stability in cognition over 12 months versus patients on placebo, that is huge, especially if we can do that in better than 50, 60% of patients. That is a monster drug that's going to help millions of people, not just in the US, but around the world.
So, and someone is, a viewer is asking, what is the target on what protein that you feel your drug candidate is aimed at? So specifically, you know, what is the protein? Is there a protein that you're aiming it at? I mean,
oh yeah. Okay, now you're starting to speak my language.
Yeah, exactly. Not mine, obviously.
Okay, so the target of our drug Simufilam is a scaffolding protein called filament A. We all have filament A from your head down to your toes. That's not the target. The specific target is an altered form of filament A. One of the contributions of our scientists and our academic advisors is they made the unique discovery that patients who have Alzheimer's disease have misfolded filament A. And
so something goes wrong with that protein earlier on, or just through the whole course of the disease?
Early, super early on.
So this is so is this what may lead to plaques, or is this like where is it on the?
You know, we don't know what day 0 is, and we certainly don't know what causes day 0, but it's well known by now that patients who are diagnosed, who have the clinical symptoms of Alzheimer's, typically the disease has been in place for a number of years. Whether it's 5 years or 15 years, I don't know. I don't think anyone really knows. But clearly, you know, the germ has been seeded. The seed has been in place for many years beforehand. And what we're saying is part of that seed, if you will, is a misfold in this filament A. And what our drug does is it sits on the filament A so that it is no longer distorted. It is no longer shaped oddly. It restores its normal function and shape.
So you're making the scaffolding stronger, so to speak, or you allow it to maintain its strength so you don't get that degeneration within.
Yes, I buy into the analogy. If you think of a brick house or brick building, if you remove one brick, so what? A few flies get in, right? You remove a dozen bricks, you know, maybe no big deal. You start removing 50 bricks, 100 bricks, something's going to give. Eventually, you remove, you know, 1,000 bricks and the whole scaffold folds. Something similar may be happening in Alzheimer's disease. What we're saying is, here's filament A. There's an altered form of filament A. When it's altered, it loses its function, it loses its shape. You're taking bricks away from the house. Eventually, you take enough bricks away and the whole thing collapses.
So what does this mean for the patient? Does this mean if it does indeed work and it does do that, it stops, it really reinforces the filament A protein. Does that mean this is a drug that you would stay on for the rest of your life and hope that, it's enough to stop the progression of Alzheimer's?
Yes, absolutely. Not only that, but one reason why we're developing a blood-based diagnostic is to detect the presence of this misfolded protein, because my vision is that as soon as a patient has evidence of this misfolded protein, don't wait. Start to improve your life, start to exercise, start to lose weight, eat properly, and take Simufilam early on.
So is this a drug that the people with APOE4 could possibly go on, or or those who have the early onset markers, APP, percent 1 or 2? I mean, those are the really bad markers because it means you're going to get it, right? So is this something that could be good for people who don't yet have any symptom, but they know that they have a higher chance just because there's a genetic predisposition?
Okay, so once again, it gets really complicated from here, but you're asking the right questions. What is the genetic component of Alzheimer's? Well, I think, if you really look at the data, you have to say it is not a gene-based disease. Well, it's gene-based, but it is not an inherited disease. And yet, if you have a history of Alzheimer's, you probably have a higher percentage. So you can't rule out the genetic component. All this to say that geneticists haven't figured it out yet. Let me give you an example. Many years ago, I started a genetic research company that has gone on and done very, very well for itself. But way back, and I want to say this was about 25 years ago, if not 30, we actually dissected the presenilin, the PS1 gene, and the PS2, and the APP gene. And we had some really good technical success in mapping out all the various biological pathways. And here we are 25, 30 years later, and we still don't have a drug around those genes. So we believe more in the biology of disease than in the genetics of disease.
So we're getting a lot of questions around the blood tests. I think you called it Sava DX. Is that correct? Diagnostic?
SavaDX.
Yeah,
Yes.
SavaDX.
Easy name.
Okay. What's the timeline for that? And are you going to market that for doctors to use as indicators, to see if this is the right thing to prescribe? How does all that work?
So we don't have all the answers up front. I want to be clear about that. And it is still investigational, but I'll tell you what our vision is. So my vision is a blood-based diagnostic where you go in and, you go in once a year or whatever, you get your cholesterol test and you get this and that, whatever. Included in that blood work is a test for the misfolded filament A, which is SavaDX. And if you have it, then you can sit down with your family, with your physicians, and kind of figure out, okay, what should we do, if anything? And some patients may even say, you know what, I don't even want to know. Until you tell me there's a treatment for Alzheimer's, I don't want to know. And you have to respect those opinions. But essentially, our vision is a blood-based diagnostic that detects the presence and the severity of Alzheimer's disease years before the memory loss, years before, who are you type questions and how do I find my way back home from the supermarket?
Yeah, well, and I think it's interesting. I'm curious, Remi, just why you think. I mean, you said a couple things that were interesting about how there's not going to be a silver bullet, and why. I mean, you've dedicated now, I guess, more than a decade of your life looking to help people who have Alzheimer's. Why has it been so hard? Like, is it like some people say we're going after the wrong target? Like, why have we not been able to find the right treatment or drug for Alzheimer's disease after so many years?
Deborah, it's just a hard problem. I'll throw out a number, 1 trillion. 1 trillion is the number of cells that are estimated to be in the average brain. And you're going in there and trying to fix, maybe a small fraction of the trillion and leave the other, 99%. It's a hard problem. There's just a lot of biology that we fundamentally do not understand. So I would say at the headline level, I would say it is just a hard problem. Possibly, understanding the brain may be one of the hardest problems at every level, not just at the level of biology, but at the level of philosophy, theology, sociology, you name it, it's a hard problem. I think below that, the bylines would be, there has been such a push to promote the anti-amyloid hypothesis that, frankly, a lot of the innovations just...
So where are you on that? Do you think that's the wrong target? We shouldn't have, or do we not know? Should we have not invested so much in the anti-amyloids? I mean, where are you in?
I tell you, I like the way NIH is supporting the field. What they concluded some years back is, look, the anti-amyloid hypothesis is for real. It's like climate change. It's hard to deny, right? However, it may not be the final answer. It may not be the answer at all. We don't know. Companies have run something like 29 large, long phase 3 studies around the anti-amyloid hypothesis. And to my knowledge, only one has shown a little bit of evidence for efficacy. That's the Biogen study. So there's more to it than the anti-amyloid hypothesis, which is not to say it's wrong. It's just that, like anything else having to do with biology, it's the reality is probably more complicated. And we just need to expand our horizons around the types of innovations we're willing to take on. And this is where, excuse me, I started to mention the NIH. NIH has a superb program that essentially they allow 1,000 flowers to bloom. They will fund 1,000 flowers and say, you know what? We don't know which ones are going to work. But if we let 1,000 flowers bloom, maybe a couple dozens are going to show real evidence of efficacy. And of the couple of thousands, a couple dozens, maybe 3 or 4 are going to turn into real drug candidates. And out of the 3 and 4, a couple may get approved. And that's where we come into play.
So in the early data that you're getting, is there a reason why this drug wouldn't work on people? And do you know, is there something like, I mean, it's really interesting right now to look at the section between lifestyle prevention and, treatments and drug therapies. So I'm wondering where you are on that. And if there is like maybe people who have more comorbidities or something that, could make it more difficult for a drug like this to work.
No, I think the science behind our drug, the underlying science really speaks to anyone who has misfolded filament A in the brain. So in that regard, the drug should work with all patients who have misfolded filament A.
But just to be clear, and that's pretty much everyone who has Alzheimer's?
Yes, but here's the difficulty. Not everyone who enrolls in an Alzheimer's disease actually has, there are different types of Alzheimer's. So not everyone is going to have the misfolded filament A. You may have what's called vascular Alzheimer's disease. You may have symptoms of Alzheimer's, but in fact, you may have had a stroke. Patient may be in deep, deep depression and show all the symptoms, all the behavior of Alzheimer's, but in fact have nothing to do with Alzheimer's. So, sometimes people come up to me and go, Remi, I don't get it. It's a crisis out there. We're all looking for answers. Why are you doing 1,000 patient studies? well, we have to do 1,000 patient studies because we know that by enrolling 1,000, maybe only 6, maybe only 700 actually come in with the Alzheimer's disease that we can treat. And unfortunately, the other 20, 30% may have things that have the look and feel of Alzheimer's, but are not, in fact, Alzheimer's disease.
So how long have we known that the filament A is such a thing? I mean, is this a newer discovery, or is this something we've known for a long time?
Time is relative, right? No, but this is one of our scientific contributions to the field. This was an observation made, I want to say, approximately 12 years ago. So yes, in science, 12 years is nothing. That's very, very, soon. So it's a recent discovery, and unfortunately, drug development is a slow, and it should be slow. There can't be anything, with the exception of things like COVID. There should not be, rapid drug development, because then you put people at risk, and it's not good for anyone.
So we got a question about, I mean, you said yourself you're a small company or small biotech company. Are you going to partner with pharma? Like, what if in phase 3 shows that, yeah, this really works? How do you bring it? Like, what's the next step? How do you scale?
We are, our specialty is innovation. Our specialty is research and development. That's what I've been doing for my entire career, for 30 plus years. We're not a sales and marketing shop. I wouldn't know how to sell a drug if it hit me in the head. So at the right point, yes, we probably will hand this drug over to someone who has all the infrastructure in place, who specializes in distribution and getting the drug from the warehouse to the patient. That's a whole separate set of complexities and specialties. Frankly, I don't even understand how that all works. We're just focused on the science and the clinical development.
So one of the unintended consequences that we have had at being patients is because we're explaining the science to people. We often get people holding up their hands saying, how do I get into this study? How do I find this, the right trial? Where should we direct people if they're interested in inquiring more about your phase 3 trial? And you said towards the end of 2021, just to be clear, like mid to end, right? We're a few months out now.
Excuse me. I drank some water.
Sorry, I made you talk too much, probably.
Yes, we're still a few months out. As I mentioned, we are increasing the enrollment in, our open label study. But the real study that, if I had a parent or a close one with Alzheimer's, I would want them in the phase 3 study. That's really the go-to study. And those will be initiated, we think, sometime around September, October or so. Meanwhile, right now we have 9 clinical sites around the country in Arizona, Ohio, Florida, Oklahoma, various places. I think they're posted on our website. They should be. If not, I will check right after this. That is the best place to start. I will say, please don't call our scientists. I'm not saying this to be mean. It's that we really are getting dozens and dozens of phone calls a day and emails, and we can't handle it and do our work at the same time. We're here to help, and we will help. But the proper way to help is to really refer patients to the clinical sites, and they can answer the specific questions of, does my father qualify, or does my family member qualify to enter such and such a study?
So, so when is it that you will have the phase 3? I mean, you still need FDA approval, right? Or
correct, yeah.
So you got to wait for the approval. Once you get the approval, you can go into phase 3. Then phase 3 will last, you said, I think 6 to 8 months. That's the shorter study, right? And then another one will be longer.
So there are 2 phase 3 studies. One will be approximately 9 to 12 months. The other will be approximately 18 months.
Okay.
That's for patient enrollment. And then you need to analyze the data. You need to see, you know, what does it all mean? And then you package it, you send it to FDA, and then FDA typically takes with breakthrough therapy or the right designation, they typically take 6 months to review the package, and then it's in the hands of patients.
Okay, and just so that people are clear, I mean, you know, Cassava Sciences got a lot of press lately because I think there was a lot of investors very bullish on the early data. What is it? I mean, you know, let's face it, phase 2 is early days, right? We're not talking about like something that is like, oh, I know this is going to work. This is like promising to advance to the next stage, right? But
right.
In particular, like specifically, what did the data say that made you excited? I mean, you've been doing this for 12 years. What is it, you know, what specifically were you looking at?
So it's a great question, but the way I want to answer it is that it's not any one specific data. See, that's the difference with our approach. It's the consistency of data from the basic research to the animal data, to the CSF biomarkers, to the early cognition, to the open label study. There's a consistent theme that, in my opinion, the world of Alzheimer's disease R&D has never seen before. What we're used to seeing is, oh, here's a drug for Alzheimer's. Here's a study that shows it goes in the brain. Let's move on and repeat.
Oh, sorry, that's my dog. I knew this was going to happen. I'm muting.
Okay. I'm going to assume you can hear me. So what's different here is, again, the consistency of data, all the way from basic research to human data and early evidence of cognition. You can actually connect all the dots and reach some sort of aha moment to say, wow, if the data replicates in phase 3, this is for real. And I think that's what a lot of you know, frankly, what a lot of people are seeing, that you can connect the dots, that we're small but very transparent company. We have a lot of peer-reviewed publications. We have more coming out. And if someone is so inclined, they can actually sit down and try to understand all this stuff and reach the conclusion that, yeah, this is really different. Not only is it different, but these guys are actually using hard evidence, biomarkers. Biomarkers don't bluff. There's no placebo effect with biomarkers. If you suppress biomarkers, you will improve health.
So just to end on, and thank you so much for this time. I think it's, you know, really interesting, and of course we are all hoping that, you know, any company will solve part of this mystery or the whole mystery behind Alzheimer's. I have to ask you, just curious, you know, you've dedicated a lot of your life to Alzheimer's disease. As a scientist, why? I'm curious, you know, what the reason was behind that.
It's what we do. It's what curious people do.
So you haven't been, have you been impacted in your family by Alzheimer's? There's
some of that too.
Yeah. It's hard actually today to find someone who hasn't been, right?
Yeah, exactly.
It's such a, so Remi, thank you so much for your time. It was a really interesting conversation. And of course, we wish you all the luck in the world, as we do of any scientists looking to solve this mystery. But especially, you know, thank you so much for your time and to take the time to really explain it all to us. And we look forward to hearing about the results to the phase 3 clinical study. And of course, if you missed any of this interview, we always post it on beingpatient.com. You can make sure to sign up for our newsletters. We, you know, always keep you abreast of upcoming talks, whether they're from scientist perspective or patient perspective. We feel like Being Patient is a great opportunity for people to see Alzheimer's from all sides and all dimensions. So thank you very much, Remi, for joining us, and we wish you all the luck.
Thank you, Deborah, for having me.